Hello judges, my name is Sidharth Srivastava, and today I will be talking about my research project, the effect of a high surface area anode design on the power generation of a pond sludge microbial fuel cell. The world is facing an energy crisis. As a result, new renewable technologies are needed. One of the biggest environmental challenges today is battery waste. Batteries use toxic metals like lithium, nickel, and cadmium, which are deposited in the ground after use. To remedy this, renewable, portable energy generation must be developed. Enter the microbial fuel cell. A microbial fuel cell is a type of fuel cell which only uses bacteria and sugar to generate electricity. A microbial fuel cell, or MFC, consists of three main parts: an anode, a cathode, and bacteria. MFCs generate electricity by using bacterial anaerobic respiration. In aerobic respiration, oxygen is used as the final electron acceptor. However, in anaerobic respiration, the final electron acceptors can be a variety of substances. Bacteria are able to form a biofilm on the anode of an MFC. Certain species of bacteria, called exoelectrogenic bacteria, can conduct the excess electrons from anaerobic respiration out of the bacterium using nanowires or membrane proteins and into an outside substance, such as the electrically conductive anode. The excess protons or hydrogen ions from the reaction move to the cathode through the chamber of an MFC, as there is now a potential difference between the anode and the cathode. Here is a diagram of how an MFC works. The electrons can move from the anode through a circuit and into the cathode. In the cathode, another half reaction occurs. The cathode is facing the air, allowing it to react with the air. Oxygen is reduced using the hydrogen ions and the electrons flowing through the circuit. This is accomplished using the help of a catalyst, such as platinum. While promising, MFCs suffer from lower power outputs. To increase power output of an MFC, biofilm formation has to increase, as more bacteria will result in more electrons flowing through the circuit. In this project, increasing anode surface area was hypothesized to increase the amount of biofilm formation. To increase anode surface area without making the MFC bigger, a design inspired by mitochondria was used. To increase surface area without increasing volume, mitochondria used folds called cristae, as you can see in the image. These folds can be used in an MFC design to increase surface area without increasing volume. This project tested whether this modification had a significant effect on MFC power production. In this project, two MFCs were 3D printed and assembled. They were filled with pond sludge, which is a good source of exoelectrogenic bacteria, sugar, and a medium solution. They were put under load and run for 24 hours. An Arduino and a sensor were used to measure voltage, current, and power of these two cells. Several assumptions were made in this project. The first assumption is that the amount of bacteria in sludge in both cells would remain constant. This is because the sludge was taken from the same location. The second assumption is that operating conditions will remain similar. This is because the cells were nearly identical and were run in the same room. The final assumption was that the measuring devices are accurate. The measuring devices were tested prior to their use on the cells to verify their accuracy. First, there were a few design considerations when constructing these two MFCs. First, the cathode had to be exposed to air, but it had to be watertight to avoid letting out liquid. To waterproof the cathode, a gas diffusion layer must be used. GDLs are usually made of polymers such as PTFE. However, I could not dissolve these polymers in my lab due to safety concerns. I reached out to a professor at Penn State, Dr. Bruce Logan, who was able to help me find GDL material, silicone spray. This was one of the biggest challenges facing this project. Secondly, the cells had to be watertight. Rubber gasket material was used for this purpose. Finally, a medium was required to sustain the bacteria in the cell. Here are my materials. Here's the equipment I used. And here are my facilities. Now I will talk about my methodology. Two MFCs were constructed to test the new anode design. Both used the same chamber, cathode backing, cathode material, and anode material. The anode material was graphite felt. The cathode material was carbon paper with 40% PT catalyst. The anode backings, cathode backings, and chamber were modeled in Autodesk Inventor. These models were then 3D printed. The anode material was glued onto the anode backing with wire in between. The cathode material was sprayed with silicone spray for waterproofing and pressed on to the back with wire in between. The cells were sealed using rubber gaskets and held together using threaded rods and wing nuts. A power measuring circuit was constructed using an Arduino, an INA219 current and voltage sensor, and an SD card reader. The Arduino was programmed to read current and voltage from the sensor and multiply them to find power. A buffer solution consisting of sodium phosphate and biological oxygen demand pillows was prepared to help maintain the bacteria. Pond sludge was collected from the Brookdale Community College pond. The cells were loaded with pond sludge, buffer, and glucose. They were wired to the measuring circuit and measured under constant 10K ohm resistance for 24 hours. The data was then saved to an SD card, and a sampling routine was written in C++ to find 24 regularly spaced data points from the file. These data points were used for statistical analysis and graphing. Finally, the cells were disinfected using bleach and disposed of. Here are photos of my models. Here is a video of the models coming together to form the final cell. Here are photos of my procedure. And here are photos of my experimental setup. On the top, there are two bar graphs of both levels of the independent variable. The dependent variable in both graphs, power output, is graphed against trial number, where each trial was one of the 24 sample data points. On the bottom, there are two scatter plots with 3 standard deviation error bars of the same data. The graph on the top compares the mean power output of the flat anode to the curved anode. The two graphs on the bottom are voltage graphs over time for both cells. As evident, the voltages stayed quite constant, save for a small blip at the end. Here is my summative data table. For statistical analysis, a t-test was used, as there were two levels of independent variable. The t-test was two-tailed, as both a positive and negative significant difference were accounted for in the hypotheses. The t-test was independent, as the subject in each level of the independent variable were unique. The alpha for this test was 0.05. The p-value for this test was less than the alpha, so the alternate hypothesis was supported and the null hypothesis was rejected. The power output of the flat anode MFC was on average 48.2% higher. This is possibly due to decreased biofilm formation on the curved anode. The curved anode could have made it harder for bacteria to form biofilm on the surface due to its shape, or it could have had less contact with the sludge. While the power output did not increase, this project did provide valuable insights into the function of MFCs and is a good baseline for future projects. There are many future studies and improvements on this study. Wastewater could have been used instead of pond sludge, leading to higher power outputs as it contains more species of anaerobic bacteria. The cells could have been purged with nitrogen to remove trace oxygen, allowing for complete anaerobic respiration. The cells could have been two-chambered, allowing for independent observation of both half reactions and the use of a proton exchange membrane. The anode material could be changed, as porous carbon paper or cloth may allow for more biofilm formation. A three-dimensional anode could be used, giving much more surface area for biofilm formation on all sides. Finally, nanoparticles could be used to allow bacteria to attach to the electrode surface better. Finally, here's my bibliography. And here are appendices with miscellaneous info and experimental design. Thank you for watching.